I just mentioned in my previous statement that usually a risk is associated with a negative component, which is not really 100% true, because once I identified a risk and I have a risk under control, it may mean that this even has a positive aspect, because I can rest assured that I have my risk under control, which may be really positive for the overall outcome of my trial. Or I may be able to detect an assumed risk, such as an under-enrollment, which all of a sudden turns into a rather quick enrollment, which helps me to terminate a trial earlier, because I already identified and enrolled the expected number of patients for my trial. So risk is not necessarily always something negative. It can also have a positive component. So the risks itself, also come with three dimensions, three areas, three facets, and those are the probability or the likelihood that something is going to happen. The second is the impact. What is the impact of a particular risk on my study outcome, on my budget, on the quality of my data? And the third dimension is the detectability. And just to give you an example from a real clinical trial, a protocol required in an anti-infective drug that the urine in the patient should have a certain number of organisms, and if the number of organisms is lower than that, the patient was not supposed to be enrolled, and if the number of organisms met a certain criterion, then the patient was okay to be enrolled based on the number of organisms. Now, the impact of enrolling wrong patients into the trial is that the trial may not meet the number of patients eventually required for a proper statistical analysis, thus resulting in a trial to be repeated. What is the probability to detect these abnormalities in the trial? The probability that this happens in the trial is rather high, because usually these trials require between 800 and 1200 patients and a lot of different sites, and the likelihood that a site enrolls the incorrect patients is rather high. So someone needs to take a look at that. And what is the detectability in such a case, in such a scenario? It is rather high, because if I take a look already at the first patient or second patient enrolled into the trial, I would probably, hopefully detect that the patient incorrectly has been enrolled, and can then take appropriate corrective actions by contacting the sites and referring to the protocol requiring a certain number of organisms in the urine, and thereafter hopefully the patients enrolled are the appropriate ones rather than incorrectly enrolled patients. In the examples that I gave, I pretty much looked at the negative sides of risks and risk management. But there's also a positive component that one should not necessarily forget about. So if I determined already what the risks are in my trial, in my program, it is certainly beneficial to have that in place, because once I see certain signals, I have that risk already under control and can react appropriately. Or even further, on the positive side, if I expect a certain number of patients to be enrolled in a particular time frame, for instance 50 patients per month, and I realize by looking at the risk components that I enroll already 100 patients per month, this could be a very positive aspect, because I can terminate my trial early. I can finish the trial early and have my data and my results available much earlier than originally anticipated.